Hey guys, I took it upon myself to make this video. I'm sure you guys are excited about this. Since we've missed so many days, I want us to stay on task so that the next time I see you, we could just move right ahead. Lucky for you guys, I brought home an expo marker and I had this whiteboard at home. So what I want you to do is take out your note packet from this week. It looks like this. Okay, and remember you could pause this video at any time to go ahead and get whatever material you need. So we're going to turn the page and go to number 2 because we've worked on section 1 this week in class. So section 2 looks like this. And the question is, how does genetic engineering affect us? So we're going to look at 3 different ways that genetic engineering affect us, and throughout this I'll give you some examples and I'll try to make this video as quick as possible. You have all your stuff. Let's get ready and it's time to go. Here we go. A. This is number 2. How does genetic engineering affect us? A. The first way is that it is used to make genetically modified plants for use in agriculture. Examples of that is that crop can become resistant to diseases and to pesticides. So that's one way that we're using the technique of genetic engineering in our society today. So remember, genetic engineering, we use one genetic information and insert it into another organism in order to get the trait or the characteristics that we want. I want you to take a second and turn to the next page to number 3. It looks like this. My chart is a little off, but in your notes it says potential benefits for GM plants. So it looks something like this, your number 3. So in this section, we're talking about what are the benefits to genetically modifying crops. So that number 1 slot is that now we have pest resistant. Filled in for you is number 2 and 3, where it says herbicide resistant, cold tolerant, and the 4th one is to create drought tolerant plants. The arrow continues to show you that this technique, genetically modifying crops, improve our farming. You have to fill in the 2 charts over here, and this technique provides us with, fill it in, cheaper food, and also our ability to make more food. And as a result, this technique reduces world hunger and improve world health. You also have this little circle here. Let's talk about concerns. What are the concerns we have about genetically modifying plant? And one concern we could put here in this section is that it create a super weed, meaning that we could genetically modify something that we don't have anything out there to get rid of it. And it can become an invasive species where we can't get rid of it. And that could be a problem. So that's problem number 1. Problem number 2, or concern number 2, is that gene from a nut can be put into another plant. This could be dangerous to people with nut allergy. Right? So that's one concern. If we're using the gene of a nut in another food, will people know that it's from a nut plant, and will it have an allergic reaction to people who have nut allergies? The last one, number 3, one of our concerns with genetically modifying crops, let's write it down. Is it ethical to eat a food containing gene from something or someone where someone would not eat it due to religious or health reasons? So we have to keep in mind, if someone for religious reason or health reason don't eat a particular product, if we're using that gene into a food, will they know about it? Will it have an effect on them? So for ethical reasons, that's something to consider. Okay, so now you could go back to number 2. Okay, let's go back to the first page. B. Another way that genetic engineering is affecting us, now we're creating these transgenic organisms, meaning that they're living things that have genetic information inserted in from a different organism, where two organism genetic information are combined. So for B, we're going to put that B, we're using this technique to make medicines, or make vaccines, excuse me. And the way an example of this is that we're making harmless form of a pathogen. So that's a pathogen is a bacteria or virus, anything that could trigger your immune system. So we're genetically modifying vaccines so that they could be harmless, so we can fool our immune system in order for us to trigger an immune response. C, genetic engineering affect us because we're using recombinant DNA. And the way we're using it is that it may someday help treat a variety of diseases. So we saw this in the video in class, that Do Now video, where we're seeing that through gene therapy, we're able to manipulate an individual's gene to treat a disease. So by mapping all the genes, we're able to locate maybe genes that are more likely to create a disorder and be able to remove them. Okay? And so on the last page of the notes, it's really self-explanatory. It talks about problem with this technique, gene therapy. Problems with it is that, do we have enough scientific evidence to make sure that this is effective? Also, it's very expensive, this method of gene therapy. Should it be used for treating serious diseases, enhance physical appearance, enhance maybe athletic ability, enhance intelligence? Should it be used for this? So there are questions in whether we should use gene therapy and what we should use it for. Also, for ethical reasoning, is it right to alter your gene? Should we genetically engineer cells of a zygote, a cell that's being developed into a child? Okay, should we interfere with evolution? That's a question. Genes that are damaged under some condition may be advantage to others. We don't know if having an altered gene will be an advantage to our overall race. All right, and also, could gene therapy be used as a new form of terrorism? So these are all problems concerning gene therapy that you have listed out on that last page. So here, just quick notes, guys. So fill in your note packet. And also a reminder to finish your lab on genetic engineering that we did in class. Those last questions on the back. You can get those for you. The last questions, you need to answer the following analysis questions, numbers 1 through 5. Number 1, I'm just going to read through them for you because I know for some of you guys that's helpful. So number 1, why must the insulin gene have a sticky end on both sides to fit into the plasmid? So remember, the plasmid was the yellow strip, the insulin was the blue strip. Why did it have those free-floating sticky ends? In order to fit with the plasmid. Check your notes in your lab book for that one. Number 2, predict what might happen if a circular plasmid had more than one cut site for the restrictive enzyme. So think about that yellow strip. What problems, or what do you think would happen if we cut it not only in the one spot, but cut it in different places? What do you think would have happened? Number 3, why would you encounter problems if you cut the insulin gene with the BAM, that's the restrictive enzyme, that's the second one that we use, and the plasmid for the EcoR1? So what problems would you have if you cut the insulin, which is the blue, with the BAM? Let's see the BAM look like that. Okay, that's in procedure number 2. And then what would happen if you cut the plasmid, the yellow one, with the EcoR1? What problems would happen? All right, so just go back and look in the packet for that information. 4, what would the bacteria and all its descendant produce after it has been genetically engineered, after it has a genetically engineered plasmid incorporated in it. So we made those rings in class, the yellow and the blue ring. So once I put that ring back into the bacteria, what will that bacteria be able to produce? That's what that question's asking. Give some examples. Those are the 3 blue strips. Those are the 3 examples of things that can be made. Number 5, why are the restrictive enzyme, the ECOR1 and the BAM1, made better for genetic engineering and constructing plasmid than HAY 1, 2, 3? All right, so once again, let me just rephrase that again. Why are the restrictive enzyme from procedure 1, the restrictive enzyme from procedure 2, why are those better for genetic engineering than the enzyme we use in procedure 3? The hint is to look at the ends, look at how we cut them, where they would be cut, and that will explain to you why procedure number 1 and procedure number 2 enzymes are better than procedure number 3. Okay? All right, guys, that's it. Please have this done so that the next time we meet, we can move on with this material. Thank you. See you later.